Hello and welcome to Talking Pharmaceuticals, an interview series brought to you by Making Pharmaceuticals and Distributing Pharmaceuticals. I'm Victoria Emerton, Conference Director for the events which next take place in April 2025. And in the lead up, I'll be joined by experts from UK Pharma to share insights, opinions and practical advice, providing a fresh perspective on key challenges and opportunities in the industry and to look ahead to their involvement in the events. I'm delighted to be joined today by Peter Deegan, Digital Quality Lead, PACE Automated Packing Capability at Automated Industrial Robotics. Welcome, Peter. It's lovely to talk to you today, and thank you for sparing the time for talking pharmaceuticals.
My pleasure. I'm glad to be here.
So Making and Distributing Pharmaceuticals has an awards and dinner that takes place on the middle evening of the events, and Automated Industrial Robotics have been shortlisted for the Innovation in Manufacture Award with the entry PACE Automated Clinical Pharmaceutical Packaging Capability, and we're going to be talking a bit about that today. So, Peter, could you start off by telling us what is PACE technology and how is it enabling zero waste, zero error, just-in-time pharmaceutical manufacturing and supply?
Yes, well, thank you very much. And we're also delighted to be on the shortlist for the Innovation in Manufacture. It's exactly where we want to be in innovation. PACE was born from a catapult project which started in 2018. Sponsored by the founding partners AstraZeneca and GSK, to learn about innovation through technology to deliver an industry problem that is really very prevalent in clinical supply. In clinical supply, we usually make to campaign. We make lots of bottles, label them, and make sure that we've got enough stock so that when we start the clinical trial, we can provide them to the clinics. The great challenge that we have in clinical studies is the rapid amount of change. So what happens is usually labeling changes happen or study centres happen. Study centres change their protocols. Indeed, you can't recruit as many patients as you need. So there's a high waste that we really want to try and overcome. And the innovation project was really designed to say, well, how can we do that? How can we do a just-in-time manufacturing capability to supply clinical trials on a make-to-order capability? It doesn't really happen like that normally. And with a make-to-order, you can get a signal from the clinical investigator at site. They'll click on their iPad, can we have one more bottle, please? And this system was hoping us, through innovation, to fill it, seal it, cap it, label it on the line as a batch size of 1 to go to that named patient. It's never happened really in industry before. And that's really what PACE was trying to achieve, is develop innovation technology to deliver to a just-in-time. And there's 2 components to that. Number 1, there's the technology, the machinery, and that's where we engage within Automated Industrial Robotics. They're one of the leading robotics manufacturing capabilities. They supply FMCG and pharma to say, how do we do this? How can you think differently? And they said, well, automation, sensor-driven automation. You can design the human QC checking out, because then you can completely bound that quality system and go to zero errors, because it's fully censored, fully checked by the machine, fully verified in a validated state. The second thing is that the amount of data that is consumed when we're checking information, when we're checking the back sheet, when we're checking the registration status to say, is that okay, can be automated as well. So the machinery, but also as well the data. And that's really what we tried to marry together to build a just-in-time capability.
It sounds fantastic and incredibly responsive as well. Can you give us some detail on how PACE works?
Yeah, so the other area of concern that we've got in pharmaceutical manufacture is cross-contamination. The GMPs are very much driven on single product manufacture to avoid cross-contamination. When you're running a clinical trial, You've got the active, you might have a placebo. If you're running such as a cancer trial, you might have a comparator against which you're doing it. You've got other elements of the kit that you've got to include. And one of the challenges that we have in that is manufacturing consistently. There may well be printed and packed on a separate line. There may well be subtle differences. So what PACE did through industrial robotics technology and design is they developed individual silo-based filling sealed capability. And the prototype, we've got 4 filling heads. We can have 4 completely separate products in hoppers, in bulk tablets, for example, that they can dispense into a bottle using a sealed technology, seal it and cap it right next to other products. And the beauty of that then is that those bottles, with different products in them, go down the conveyor belt to a single inline printer. So that's the first differentiator, multi-product fill line. Again, much faster, much quicker, much better security. The second information that we can utilize is labeling. We predefine the labeling parameters. In clinical trials, you've got what's called the master data label, the study master data label, okay, which is approved by the regulatory authority. It's got the safety information regarding the product, etc. That can change very quickly. So we create the study master data label in a template, in a printing system. That's there as a mask. And what then PACE can do, you've got these 4 bottles on the line, each with 4 different products. It's got a laser etch on the bottom so that the sensors know which bottle is where. You present that to the labeler. It prints off with the variable data, the kit ID, the lot information, and the safety information onto that label. Before it's applied to the bottle, it's then printed. Before it's applied to the bottle, a camera takes a photograph of the label and it goes back to the label verification system and verifies that that is the actual data. Traditional camera verification systems usually just check the barcode, but this checks all the data. So there's 0 error. And that's before it's applied to the label. Once it's applied to the bottle, then it's a second camera verification for alignment. You can have multi-wraparound labels, etc. And then it's labeled. But the beauty of the labeling system is you only label it on the line. It's only printed on the line when you need it. So that's when you can go down to a batch size of 1. So that's how PACE works. You've got hoppers with different products in. You get an IRT order. It puts a signal to fill that bottle and cap and seal it. It goes then to the labeler and then labels it. And then it's stored in a modular storage system ready for QA review. So then the QA review comes into play, where we estimated and analysed within AstraZeneca and GSK, for a standard batch review, there's about 1,200 data points. And you think, okay, that's got to be checked by humans. The biggest challenge is that what we found is that those 1,200 data points can actually be spread across 23 different electronic systems that you've got to log in, check in, and look to. We say we've digitalized our data. The way that we put it on the project, we've electronified it. It's in a PDF, it's in a Word document, it's in an Excel or as a table. It still needs a human eye to check that. Okay, and we've had recent times in the press where we've had recalls down to human errors, wrong product name on a batch of product. So what we did with the PACE system is we tried to think differently and say, well, how can we design out that fault as a capability? So what we do is we pre-flight check the data. We've got the registration data, we've got the lot information, the expiry date, all that information. It comes into the system. It's come directly digitalized from the original source system. A lot of people talk about artificial intelligence at the moment, but this is basic machine automation. What was the specification? The machine checks the result. Okay. Humans are making errors because we're very highly qualified, we're very highly trained, but we still know that human process capability is not infallible. So the machine checks it with 100% accuracy. And in sometimes you can't even pack the batch if the pre-flight data isn't there or isn't viable. The reason being is that just in time is not really fast packaging followed by a long wait for human QC checking. We had to combine those two elements together. So that's the final beauty of the PACE system, is that the data is digitally assessed and verified. It brings it to what we call this QP dashboard, which is all the information which the QP or the QA officer can review. It's digitally verified. We know that current QP legislation and QA legislation requires a human review and sign off. We've managed that interface. There is a great concern and feedback from inspectorates that the large number of errors that are happening today are between that human and machine interface. But we've been able to verify that the machine is immeasurably better and more accurate at checking the data and verifying that data. And what we've started to use is a phrase called quality verification. So let me explain that. Today, with the GMPs, you read the GMP texts and guidances, and they're all about the human shall check this, you shall double check and verify, etc. Follow this, sign that, etc. That's quality assurance. But what we're saying now, and PACE has been able to prove it, that we move towards quality verification. A lot of people talk about release by exception. I'm saying release by verification. The machine has verified better than the human can that the data is sound, the data is attributable, the data is there, and the data is verified. So that's when you can really rely more on the data, because the machine's done it through standard automated checking. And that's how we built the machine together with the data together to get just-in-time packaging.
That sounds amazing. A little bit big brothery, maybe, but all to the good, I would say. And AI is certainly something to be embraced. And we're going to be covering the uses of AI in pharmaceutical technology and manufacturing at the event, actually. So how is PACE improving manufacturing accuracy and reducing waste and rejects? As you say, it's well documented that speeding anything up, speeding up manufacture has a higher risk of more errors. So how is that being minimized by this system?
Well, that's a really good point. We did look at just in time, and we did look at human processes involved in that. And there are well documented psychological and texts in the media about human process checking capability, human faults. And they do know that if you speed up activity or put more volume to a human, they will make more mistakes. The challenge that we had with PACE was that we can pack one bottle as one batch size at a time in minutes. So if you think about that, you could arguably pack, label, and QP certify 1,000 batches a day of one bottle. Today, traditionally, we may well make 1 to 5,000 bottles in QP at once. So you can see the issue that I can't humanly check 1,000 batches with that level of accuracy that quickly in the same time frame. So we had to really turn it round and think, okay, how can automation help us speed up but actually improve? And there's great technical analysis that we did to be able to say, okay, we did 20 batches of one bottle. Okay, how many errors were there versus 20 human QC checking of that data? And it took hours and hours and days for the humans to do that, and there were more errors than the machine did. So design the human out of basic checking. Automation is the only way to achieve just-in-time manufacture in a better or at least equivalent quality verification than better than the human.
So humans aren't always necessary. I get that, and this system is showing that machines can do things better. How is PACE unique to current ways of working?
So I think there are a few things in that. Firstly, make to order versus make to campaign. It's a just-in-time capability. You only bottle it, and you only bottle enough that you're requested for when that order comes in. Okay, rather than have it all on the stock on the shelf waiting, either pre-filled, non-labeled, or labeled. The latest time that you can fill that bottle and label it, the better for the trial dynamics and the supply chain. We know that in clinical trials, labeling changes happen overnight almost. There are bioavailability changes. It's a clinical trial. You're learning about the product as you move along. You're also getting feedback from patients and clinics, etc. So the ability to do that late stage filling and labeling helps you keep current with that time. The other factor of make to order versus campaign is you just can't reduce the carbon footprint by making a load and hoping that it will be used. What happens if the study centre closes down? What happens if you don't recruit as many patients? Those are all real in the moment dynamics. So just having them on the shelf to wait is a carbon load, whereas just making that one bottle for that one patient can be very efficient in terms of carbon footprint. And in the PACE design award monograph that we put a bit about, there's some real good analysis being done on sustainability reducing. NHS has got a target to reduce carbon footprint by, I think, is it 2035 or something. You can't do that without automation. You can't do it. So that's one of the unique elements to PACE. Designing out the commoditized human QC checking. Humans are very important in the manufacturing process. You've got the qualified person. It takes 3 to 5 years to train to be a QP. But the QP has got such a high bandwidth, it can't meet just in time. That's really important. The inline labeling we think is a game changer because, again, traditionally, you make a reel of labels, you attach them to the line and you apply them to the line. You may put the barcode over on or a kit ID. Look at serialization. You don't need serialization in clinical trials, but industrial robotics are the world leaders in serialization, for example. They do it for FMCG, tax reasons, etc. They've just got that software that creates the label mask, brings that data, that serialization data in, but they can 100% verify it on the line. There's a very interesting case example when we first started using pack serialization in the European Union. The barcodes and the serial numbers which were printed at source weren't verified at source, and when they got to the pharmacies in Europe, they failed because they couldn't read the barcode. That can't happen with PACE because it's verified on the line and checksummed on the line. And I think the final unique capability of PACE, it's the only way we're going to reduce carbon footprint and achieve sustainability. It's now a huge thing. We look at the cost of pharmaceuticals, we look at the cost of trial materials, look at comparators, look at short shelf life requirements. It doesn't lend itself to good carbon footprint practice by having to destroy adequately capable pharmaceutical product. But if you use it, if you pack it only when you need it, that is a very, very clear sustainable target that you're going to achieve.
It's ticking a lot of boxes, isn't it? Cost saving being one of them, sustainability being another, and both of those are significant, aren't they? And again, we're going to be covering sustainability issues in the conference at Making Pharmaceuticals. So this is it's bang on trend.
I think also as well, it also supports point of care manufacture, the NHS and the MHRA trying to what I call the factory on the back of a truck, because you can micronize this to say, I'll only dispense when I need it and the amount that I need it and label it locally. And that's a game changer in my opinion.
Wow, amazing. So what does PACE mean for digitalisation?
I think going back to our original PACE aim, that we wanted to reduce waste and speed up the packaging capability, because again, you've got to make it 3 months in advance, order it 6 months in advance, and it takes 3 to 4 weeks to pack it and then release it. So you've got a long lead time. Your lead time can be 1 day. If you want to distribute it, it can be that just-in-time supply as well. We're also looking at the ability of drones to distribute pharmaceutical products, for example. But you need digitalisation to have pre-verified it. The risk in just-in-time manufacture and packaging is that arguably you could get an IRT order or an order for a pharmaceutical product. It could be packed, labelled, QA released, distributed, and ingested within days. Okay, so you've got to have those checks firmly in place to make sure that the safety, efficacy, and quality of the product is assured. So that's one element. Reducing the human errors. You can't speed up any faster without introducing greater error. So we did some analysis with AZ and GSK, and using great people doing great improvements and Lean Sigma techniques, we went down from 70% waste to about 40% waste. What we've proven with the data from PACE is that to get to zero waste, you can only do that with automation. You can't do it through manual means. So that's what digitalisation brings, I think. It's that if you speed up, you have to use automation. You need to design the human out for the basic checkings, because we still make errors. And if you want a zero error, zero waste capability, I know that you very rightly talked about Big Brother, but this was an innovation project to feed back to industry. Can we do this? Well, there's a prototype up in Glasgow at the Medicines Manufacturing Innovation Centre, which anybody can have a look at, and we can show you how we've done it.
Fantastic. I wish you were speaking at the conference this year, but we didn't get that over the line. You'll have to come and present to everybody in April next year, Peter.
Oh, that'd be my pleasure.
Yeah, that'd be amazing. But you've got colleagues coming to the event with you who are going to be at the event on day 2, haven't you? So anybody who wants to learn more can seek them out. Obviously, you're going to be at the dinner. My lips are sealed. I can't remember if you've won. So
well, it's about a journey, really, because I think the next phase, and again, my colleagues will be at the event where you can bump into them, but we chose the prototype on oral solid dose because it was easier to design a concept. But we also know that we've got challenges in vials, and the vast majority of supply chain now is vials, cold chain. So we're even developing PACE even more now for laser etched printing in a cold chain environment, because one of the great challenges is condensation where the labels fall off, etc. So it's a continuum as well, which is why coming back next year I'll be able to show you PACE version 2 almost.
Well, a year's a long time. I'm sure you'll be quite a different place then than you are now. So yeah, that would be a pleasure. I'll make a note. So we're looking forward to the dinner. We wish you and your colleagues luck in your shortlisting. Just to remind everybody watching this that attending the exhibition and conference is entirely free of charge, and registration is open at www.makingpharma.com. So thank you so much for your time today, Peter. We look forward to seeing you and your colleagues and everybody watching this podcast at the Coventry Building Society Arena on the 29th and 30th of April. Thank you.
Thank you. It's been my pleasure to talk to you today.